Hi everyone. In today's lecture, we will be talking about dilutions in the laboratory. Before we proceed any further, let's first talk about our specimen processing. Serum is the most frequently encountered specimen in immunological testing. To obtain serum, blood is first collected using venipuncture and is allowed to clot at room temperature or at 4 degrees Celsius, and then centrifuge from between 1000 to 2000 Gs for 10 minutes. Here's an example of a centrifuge tube showing our serum. Serum should be clear and free of any debris. Once you have obtained your serum, after centrifugation, immediately transfer the serum into another tube. You can repeat the centrifugation step for 1 minute to pellet down any cells that may have been transferred into the new tube. Here are some things to avoid during processing. First is hemolysis. Hemolyzed blood samples, like the one you see in this figure, can cause inaccuracies during immunological testing. Some ways you can avoid hemolysis is by allowing the clot to retract fully before centrifugation. Also, we need to avoid repeat centrifugation, especially if the cellular elements are still in the tube. If you need to recentrifuge your tube, make sure to remove the serum first and placing it in a new tube before recentrifugation. Next is cellular components in the serum. The presence of cells like our RBCs and WBCs in the serum can cause cross-reactivity with antibodies that we use in immunological testing. Some troubleshooting tips involve avoiding collecting serum close to the clot at the bottom of the tube and rimming the tube to encourage settling of the clot. When we rim the tube, we simply run a small toothpick around the inner edges of the tube, and this releases the clot from the tube walls and encourages settling during centrifugation. Let's now talk about different storage conditions and the use of our samples. There are 3 types of samples that you can use. First is fresh, non-heat inactivated serum. This is serum that is collected directly after centrifugation. This is usually recommended for most tests and is what is usually done in most laboratories. If you are not able to perform your test immediately, then you can refrigerate your serum. Refrigerated serum can be kept at 2 to 8 degrees for up to 72 hours. This is usually done in hospitals where we have inpatients who have multiple tests that are to be done within a time period. Next, we have frozen serum, and this is serum that is stored at negative 20 degrees or lower. You can store serum indefinitely at negative 20 degrees as long as you avoid repeat freeze-thawing processes. The more we freeze and thaw our serum, the greater chances that we destroy the different proteins and antibodies that we can find in the serum. You should only use frozen serum if there is any additional delay in testing, and usually this is done for research purposes. If you need to freeze serum and use it over an extended period of time, it is advised to make smaller aliquots of your serum before freezing them, and each time you need to use a serum sample, just take an aliquot and keep the rest of the serum in the freezer. Moving on to dilutions, we have 2 types. First is your simple dilution and your compound or serial dilutions. Why do we need to dilute our samples? In the Immunology and Serology section of our laboratories, we need to dilute our samples in order to achieve optimal proportions of our antigens and antibodies in our test solution. Prozone and postzone phenomenon may give us false negative results. Prozone is what happens when you have an excess of antibodies in solution, while postzone is when you have an excess of antigens. Dilutions must be done to obtain a zone of equivalence. And don't forget that our reactions are optimal at the zone of equivalence. Now, what is dilution? Dilution is the act of diluting something, and in the laboratory, this is usually a sample. When we dilute something, the concentration of that sample is weakened by the addition of a solvent. And in a dilution, there are 2 entities or elements. First, you have your solute, and this is the material being diluted. This can also be known as your sample. Then you have your solvent or diluent, and this is the medium making up the rest of the solution. You can also call this the diluting agent. Dilutions are usually expressed as ratios or fractions. In this example, we have the ratio 1:4 and the fraction 1/4. When referring to dilutions, these two mean exactly the same thing. Now, the top number indicates the parts of the solute or sample, and the bottom number indicates the total volume of the solution. In this example, we have 1 part solute or sample and 4 parts for the total volume of the solution. So since we know that we have 1 part solute or sample, this means that the 4 parts here are composed of 3 parts diluent and 1 part solute or sample. Here is another example. So we have the ratio 1:10 or the fraction 1/10. Again, these two mean exactly the same thing when referring to dilutions. So here, the top number indicates that our solution is composed of 1 part solute or sample, and the bottom number indicates that our solution is composed of 9 parts diluent and 1 part solute or sample. This gives us a total volume of 10 parts. There are 2 types of dilutions. The first is a simple dilution. In this type of dilution, the desired dilution is achieved in just 1 step. Then we have a compound dilution, in which the desired dilution is achieved with more than 1 step, or it takes several stages. Let's first tackle simple dilutions. Here is the formula for our simple dilution, which is dilution is equal to the solute or the sample volume over the total volume. And again, the total volume is equal to the solute or sample volume plus our diluent volume. So here we have a sample problem. For a test, 1 ml of serum is to be diluted with 3 ml of diluent. What is the dilution? So let's try to solve that. You can pause the video here while you try to solve, and unpause when you're ready to see the answer. Okay, let's take a look at the answer. If we follow our equation here on top, the dilution is equal to 1 ml, which is our 1 ml of serum sample, over 1 ml, which is our sample, plus 3 ml, which is the amount of diluent. This gives us a dilution of 1 ml over 4 ml, or a 1/4, or a 1:4 dilution. Before going any further, let us first differentiate dilutions and ratios. In the clinical laboratory, we are often tasked to make different reagents, and sometimes the instructions might call for a dilution, or they might call for us to add the reagents in a ratio. And let's see the difference between the two. As mentioned earlier, a dilution is equal to the sample or solute volume over the total volume. So in this example, 1/4 dilution is equal to 1 part sample over 1 part sample and 3 parts diluent. So in this dilution, we only add 3 parts of diluent. Meanwhile, in a ratio, it tells us exactly the number of parts for the solute and the diluent volume. In this case, a ratio is equal to the solute or sample volume over the diluent volume, rather than the total volume, which you can find in the dilution. In this example, a 1:4 ratio is equal to 1 part sample and 4 parts diluent. So it's very important not to get confused with these two whenever we make our reagents. Let's now go back to our simple dilution. Here we have another sample problem. 5 ml of a 1:10 dilution is needed for a test. How much serum and diluent are needed for this dilution? So in this problem, we know that the total volume of our solution is 5 ml, and we just need to find the serum and diluent volumes. In this case, we have to let the X be the serum or sample volume. You can pause the video here and unpause it when you have the answer. Okay, let's check out the answer. First, we have to cross multiply 1:10 and X over 5 ml. In this case, 5 ml here we know is our 10 parts of the total solution. So we cross multiply that. We come up with this equation. 5 ml is equal to 10x. Then we cancel out the 10 by dividing the whole thing by 10. And this leaves us with X, or serum volume, is equal to 5 over 10. Here, 5 over 10. And if we solve for that, that gives us 0.5 ml. This is our serum or sample volume. To get the diluent volume, we just have to subtract our sample volume from our total volume. In this case, 5 ml minus 0.5 ml is 4.5 ml, and this is the amount of diluent that we need for this problem. Let's now move on to our compound dilutions. Now, a compound dilution is a series of dilutions that are made to obtain a desired final dilution. Previously, in our simple dilution, it only takes us one step and one tube to create our desired concentration, but in a compound dilution, we have to use multiple steps and multiple tubes to obtain our desired final dilution. Also, in a compound dilution, the samples come from our previous dilutions. So in a series of 3 tubes, for example, tube 1, tube 2, and tube 3, the sample for tube 2 comes from tube 1, and the sample for tube 3 comes from tube 2. To make it all simple, all the dilutions in a compound dilution have a similar sample or solute volume. Okay, so these are the formulas for our compound dilution. So we start with the initial dilution for each tube. And this is the same as our simple dilution, solute or sample volume over the total volume. And again, the total volume is the solute or sample volume plus our diluent volume. Now, don't forget that our samples come from a previous dilution. So we also have to take the final dilution for each tube. And this is the initial dilution, this equation here, times the previous dilution, or the dilution of the sample that we are using. Also, in a compound dilution, we end up with what we call a dilution factor. And this is simply the reciprocal of the dilution. For example, if we have a dilution of 1:10, the dilution factor is 10. If we have a dilution of 1:100, the dilution factor is 100. Then we have what is called a serial dilution. This is a compound dilution in which all steps have the same initial dilution. So if we have 3 tubes, all of them have to have the same initial dilution. A compound dilution, on the other hand, can have different initial dilutions for our different tubes. To better understand the concept of compound dilutions, let's compare it with our simple dilution using this sample problem. The problem is, using 0.1 ml of serum, make a 1:500 dilution. So in this case, if you make a simple dilution, X is our diluent volume, and here we are just using the equation that we used before. 1:500 is equal to 0.1 over X plus 0.1. Again, the 0.1 here is your sample volume. So we cross multiply and we transpose and we get this value. So X, or our diluent volume, is equal to 49.9 milliliters. Now in the laboratory, this may be too much diluent to use for a single sample. And sometimes we don't have the luxury of having a lot of diluents. That's where you can make a compound dilution. So in a compound dilution, you can plan the number and sizes of simple dilutions necessary to reach the desired endpoint. So here, we are planning to make 3 tubes, and as much as possible, we want to make a compound dilution that is the least amount of steps and the most efficient way and most practical way to get our final dilution. So for example, I want to make my undiluted sample into a 1:500 dilution. So how do we get 1 and make it into 500? So maybe you can make a 1:50 dilution and then make a 1:10 dilution. Or in this case, I decided to first make a 1:5 dilution and then a 1:10 dilution and a 1:10 dilution. So 5 times 10 times 10, that will give you a 1:500 dilution. So let's start with dilution number 1, or tube number 1. Here I am getting 0.1 ml from our serum or our sample and adding it to 0.4 ml. And this gives me a initial dilution of 1:5. Now don't forget that in the compound dilution, we have to multiply the initial dilution with the dilution of the sample or the previous dilution. So in this case, our sample is an undiluted sample. So we just multiply 1 over 5 by 1 over 1, or simply 1. And that gives us a final dilution for tube number 1 as 1 over 5. Now for tube number 2, we are planning to make a 1:10 dilution. So we get 0.1 ml from tube number 1 and add it to 0.9 ml of diluting fluid. When we do this, we get a 1:10 dilution. And don't forget, we have to multiply the previous dilution or the sample dilution, which is dilution of tube number 1, 1 over 5. So we multiply these two, and that gives us 1 over 50. This is now the final dilution for tube number 2. So now we are getting closer to our 1:500 dilution. So now, for tube number 3, we will be making another 1:10 dilution. For this one, we are getting 0.1 ml of sample from tube number 2 and adding it to 0.9 ml of diluting fluid. And we end up with a 1:10 dilution. So multiply 1:10 with the previous dilution, 1:50, and that gives you a 1:500 dilution, which is our desired final dilution. Now, if we take a look at all the diluting fluid that we used, we add 0.4 plus 0.9 plus 0.9. That gives us 2.2 ml of diluent needed instead of our 49.9 ml if we did a simple dilution. So you can see here that in a compound dilution, it is more economical and it's more practical than making a simple dilution, especially if you have very large dilutions like this. All right, this time we have another compound dilution example, but this time I want you to be the one to solve it. The problem is, create a 1:400 dilution with an initial sample of 1 ml. Okay, so how would you do this? You can pause the video here while you plan it out, and once you're ready to see the answer, you can unpause the video. So do you have your answer? Let's take a look. So let's take this tube as our sample. So in order to get our 1:400 dilution, one way you can do it is by having 4 tubes. And then we have the initial dilution and the final dilution for each tube. Don't forget that our initial sample is 1 ml. So in this case, if you were to plan this out, the initial dilution for tube 1 would be 1:2. For tube 2, 1:2. For tube 3, that is 1:10. For tube 4, that is also 1:10. So if you multiply 2 by 2, that gives us 4. 4 times 10, 40. 40 times 10, that will give you 400. So we expect that the final dilution for tube number 4 will be 1:400. So let's try to solve it. So here, this would be the final dilutions. So let's see. So we add 1 ml of serum to our tube number 1. Since this is a 1:2 dilution, there should be also 1 ml of diluting fluid in tube number 1. So we multiply 1.2 by 1 because this is an undiluted sample, and that gives us a final dilution of 1:2. So we get 1 ml and transfer it to tube number 2. Since tube number 2 is again a 1:2 dilution, there is already how much diluent fluid is here in tube number 2? It should also be 1 ml. So we transfer 1 ml, and that gives us another initial dilution of 1:2. This time, we multiply our initial dilution with the previous dilution, which is also 1:2, and that gives us 1:4. So now we get our transfer 1 ml from tube number 2 into tube number 3. And remember, we planned that tube number 3 is a 1:10 dilution. So that means tube number 3 has 9 ml of diluting fluid. So we multiply our initial dilution, 1/10 or 1:10, multiplied by 1:4. That gives us a final dilution of 1:40. After, transfer 1 ml from tube number 3 to tube number 4. And again, tube number 4 is a 1:10 dilution. That means this already has 9 ml of diluting fluid in the tube. And we multiply our initial dilution, 1:10, by the previous dilution, 1:40. that gives you a final dilution of 1:400. Okay, let's now move on to our serial dilution. If you recall from our previous slides, a serial dilution is just a compound dilution in which all the steps or all the tubes have the same initial dilution. In this example, we have the problem to create a 1:10,000 dilution with an initial sample of 1 ml. So let's see how we would do a serial dilution. First, we take our undiluted sample and we will plan to create 4 tubes or 4 steps for our serial dilution. Since it's a serial dilution, all the tubes have an initial dilution of 1:10. Let's see the final dilution for each tube. So here, our tube number 1 will have a final dilution of 1:10. If we multiply the two, 1:10 by 1:10, our tube number 2 will get a final dilution of 1:500. Tube number 3 will have a final dilution of 1:1000. And tube number 4 will get a final dilution of 1:10,000. And that's our final desired dilution. So here, we just add 1 ml of serum or our undiluted sample into tube number 1, add 1 ml from tube number 1 to tube number 2, then we transfer 1 ml from tube number 2 to tube number 3, and another 1 ml to tube number 4. And here, we end up with our final or desired dilution. When we do serial dilutions in the laboratory, it's usually to find out the titer of a patient. When we say titer, this means, or this is an indicator of the antibody's strength, and this is the reciprocal of the dilution or the dilution factor. So when we refer to titer, this is the same as our dilution factor. In this example, if you have a 1:100 dilution, the titer will be 100. If you have a 1:10,000 dilution, the titer will be 10,000. So again, titer and dilution factor are the same thing. And also in a serial dilution, the initial dilution factor for all the tubes determines the serial dilution's folding. So when we say folding, this is just a term that we use to indicate how we dilute each tube in a serial dilution. For example, in this dilution, the initial dilution factor is 10. So this would be a 10-fold dilution. If you have a 2-fold dilution, all the tubes would have an initial factor or an initial dilution factor of 1:2. If you have a 4-fold dilution, then you would have tubes with the initial dilution factor of 1:4. Alright, now we have another sample problem. This time, you'll be the one to answer this. The problem is, make a 2-fold dilution using 0.2 ml serum in a series of 4 test tubes. Right? You can pause the video here while you try to solve for the problem. And when you're done, you can unpause the video. Let's take a look at the answer. So we have our initial sample, which is 0.2 ml. So this is our initial sample, and we need to make a series of 4 dilutions. All right. So for the initial dilution, since it's a 2-fold dilution, this means all the tubes have an initial dilution of 1:2. So when you multiply each tube with the previous dilution, you end up with the final dilution for each tube, which is 1:2, 1:4, 1:8, and 1:16. So our sample here is 0.2 ml of serum. So that means we just add our 0.2 ml of serum to tube 1, then we transfer 0.2 ml from tube 1 to tube 2, 0.2 ml from tube 2 to tube 3, and then 0.2 ml from tube 3 to tube 4. In this problem, each tube contains 0.2 ml of diluent because we are making a 1:2 dilution or a 2-fold dilution for all the tubes. Then patient serum, which is again 0.2 ml, is added to tube number 1. This is carefully mixed, and then 0.2 ml is withdrawn and added to tube number 2, and the process is continued until the last tube is reached. So how do we solve for the initial dilution and the final dilution for each tube? Let's take a look at that. Okay, this is one way you can solve it. So here, this is the long way of solving it. We multiply the initial dilution for each tube with the sample dilution or the previous dilution. So this is how we solved it for tube 1, tube 2, tube 3, and tube 4. In tube 4, we end up with a final dilution of 1:16. A shortcut that you can use is this method, which is just setting up a multiplication series. So we have 4 tubes, and each tube has a dilution of 1:2. So we just multiply 1:2 4 times. 1:2 multiplied by 1:2 multiplied by 1:2 multiplied by 1:2. So we just multiply it 4 times, and that will still give us the same dilution factor, which is 1:16. Right? When we do our multiplication series, the original dilution is just raised to a power equal to the number of tubes. So for example, if you want to know the dilution for tube number 4, the final dilution for tube number 4 rather, we just raise 1:2 by 4 because there are 4 tubes. If you wanted to know the final dilution for tube number 3, just raise 1:2, this time by 3. And that will still give you a 1:8 dilution. Alright, now we have one last problem for you to solve. This time, the sample problem is to make a 5-fold dilution using 0.2 ml in a series of 4 test tubes. We need to determine how much diluent is to be placed in each of the tubes and to calculate the initial and final dilutions for each tube. Also, if the endpoint was reached in tube number 3, what is the patient's titer? Okay, so just a heads up, for this problem, we need to make a 5-fold dilution, otherwise known as a 1:5 dilution. That means each tube will have a diluent of 0.8 ml, and the volume to be removed or discarded, or our sample, is 0.2 ml. Let's see how you would solve it. Okay, so here we have our 4 test tubes. Don't forget, it's a 5-fold dilution. So each of these tubes have a 0.8 ml of diluent already added to them. Now we add our sample, our undiluted sample. We add it to tube 1, 0.2 ml, and then we transfer 0.2 ml from one tube to the other. Our initial dilution for all the tubes is 1:5, again, since it's a 5-fold dilution. If you compute the final dilution for each tube, you would end up with 1:5 for tube number 1, 1:25 for tube number 2, 1:125 for tube number 3, and 1:625 for tube number 4. Now, the endpoint was reached at tube number 3. This means that our titer would be 125 because this is the final dilution factor for your tube number 3. Alright, so that ends our lecture for today. If you want to see more videos like this, make sure to like and subscribe, and we'll see you in the next video.